So I'm going to be talking today, and I can move this away, about some of our work that we've been doing recently, and maybe this will kind of speak to some of the questions that Safi was just fielding regarding alternative methods to look at the picture of the SRX, DRX on-off states, etc. Because we're taking the approach of using a single molecule imaging approach where we're going to be looking at regulation of thick filament in myofibrils. And just to give you an idea of where the University of Kent is, you can find that right on this little stub of the UK. London is here, and we're there in red, and you're always welcome to come and visit. Okay, so I really want to talk about dual filament regulation. So I'm going to stray a little bit into thin filament regulation and come back to thick filament regulation. So as has been described already, we understand the mechanism of contraction, the sliding filament theory, to involve myosin interacting with actin, and this is mediated by calcium. When calcium is present, calcium binds to the troponins, as you can see here. These allow the tropomyosin to move away from the actin filament, exposing myosin binding sites. Myosin then interacts with the actin filament, and then when the calcium goes away, does myosin then sit on the thick filament? And this is kind of the question that we're trying to deal with. But first of all, I just wanted to talk a little bit about how calcium binding to troponin results in exposure of these myosin binding sites. And this is some work that we've spoken to using single molecule approaches. And this is work that was conducted by a former grad student of mine, Rama Desai, in collaboration with Mike Jeeves back in 2015. And so essentially what we're looking at here in this video, which is probably what your eyes first drawn to, is two beads with an actin filament suspended between those, as you can see. So if we take a cross-sectional view, you can see here that the silica beads are actually above a microscope cover slip surface by around about 5 microns. And this allows the myosins, which are fluorescently tagged using an exchanged RLC GFP, to sit on the actin filaments in any orientation, so they can follow the twist of the actin filament. And so what you can see in this case is, for a naked actin filament, we'd expect the myosins to be able to bind anywhere. And that's what we see in the data. We see the myosins, these little fluorescent dots here in this video, excuse me, binding anywhere along the actin filament. But what happens when we regulate the system? When we regulate the system, we now have myosins that can bind to these regions, and we expect them to probably activate locally, as we see in this kind of animation here. And so what we'd then expect to see is bright clusters of fluorophores. And the brightness of this cluster actually relates to the number of myosins that are present. And so this is what we get when we actually visualize this. And you can see these really nice bright clusters of myosins binding to an actin filament that is unlabeled in this particular video. We analyze these data. So this is actually unregulated. So this is naked actin. We analyze this data using a chymographic approach, which means that we're plotting the position along the y-axis. So this is displacement versus time along the x-axis. And so you see these little fluorescent dots, and they correspond to a single myosin binding. And so what we do is we run a sliding window across the data, and we keep going, and this is where I paused it. I'll take that little slice of the data, rotate it 90 degrees, and you can see here, let me just get rid of this. So we can see here that we have one fluorescent spot and another fluorescent spot here. And if we plot those as a histogram, you can see the intensity of this spot is actually higher than this spot. And that tells us there are more myosins bound here than there are bound here. So what we then do is we plot these peak intensities as a histogram. And when we do that for naked actin, we find that the average intensity for this histogram distribution comes out according to the fluorescence of a single myosin. This tells us that on average for naked actin, unregulated actin, we only have a single myosin bound. We tend not to see any clusters binding. What happens when we use regulated actin? With regulated actin, so this has troponin, tropomyosin present, we now see a much broader distribution of events, and they center around about 7. So this tells us that on average, these clusters have about 7 myosins present, and you can see that in the raw data. So raw data here shows much brighter clusters of myosin. So what we did was in that former study was we studied the cluster formation versus a few different parameters. So first of all, we studied this versus calcium, and you can see as we increase the calcium concentration, we increase the average intensities of these peaks. And this tells us that calcium permits more myosins to bind. If we reduce the ATP concentration, so on the left here we start with 1 micromolar ATP, and we move to the right 0.1 micromolar ATP, you can see here that the midpoint shift to the right, telling us that the prolonged strong binding holds the tropomyosin door open for other myosins to bind. Finally, we also increase the myosin concentration. Again, you see as you move from left to right, an increase in the peak intensity, telling us that myosin potentiates its own binding. So we formalize that in a 3-state model, in the 3-state model, where we have the blocked, closed, and open states. So the blocked state doesn't allow myosin to bind, but it allows calcium to bind. Once calcium binds, we go to the closed state. This allows a myosin to bind, which then facilitates the binding of further myosins. Now, we wanted to know how many more myosins, so we took all of the data from these 3 acquisitions, or 3 different experiments. We fitted them simultaneously to essentially GPS the data or the size of this active region, and it tells us that binding of 1 myosin propagates the binding 11 further myosins, and that is what is behind the cooperativity that we see in myosin, in tropomyosin-based activation of the thin filament. Now, if you want to know more about that, we published a couple of papers on this a few years ago. So let's go back to our original question. So this original question based around trying to understand what is underlying this process of thick filament regulation. This might be whether these myosins actually bind to the thick filament. So we aim to, or we set out to measure thick filament regulation with high precision, and also to understand the nature of thick filament regulation. So this work really kind of started as a review paper that we published a few years ago, and this is the work of a former master's student, Lawrence Helling. And in this review, we looked at the existing data out there, and it seemed that the relaxed condition of muscle can be considered as 2 different states, essentially completely off, or as we called it, poised. And this poised state is modulated by force or phosphorylation of RLC, or myosin binding protein C. And so those shift the equilibrium from the on to the poised state. And what I mean by poised is, as we've heard earlier from Wei Kong and from Safi also, is that we're looking at trying to increase the number of heads that may be able to interact with a thin filament, so that when calcium comes along, we then go into the active state, and that modulates the amount of force experienced. So before I go into talking a little bit more about our data, I'm going to talk just a second about the potential 2 types of thin filament regulation. And again, this is recapitulating what Safi just talked about. One of those is where we have the dock situation, which is known as the interacting heads motif. And I think the next talk, Davide is going to talk about this in more detail. And secondly, there is the biochemical definition of being switched off, which we call the DRX and the SRX to date. That may change now, I guess. So here, using the MANT nucleotide chase experiment, you can characterize the early portion as being DRX and the later portion as being SRX. And these correspond to rates that are about 10-fold different. So the DRX has a rate constant of around about 0.04 per second, and the SRX has a rate constant of 0.004 per second. And I'm going to also recapitulate the statements of Safi to say that even though these thick filament IHM heads are considered off, there may be heads away from thick filament that are inactive, and Karamani in 2019 called those the refractive heads. And so it's not presently clear if the IHM is the structural correlate of the SRX, and that's something that we're all, I think, thinking about. So going on to the experiments that we performed, we wanted to look at the dynamics of the ATPase within the myofibril. And this is the work of my two master's students at the time. Matt has now gone on to take this further as a PhD student. And essentially what we did was we took a glass cover slip, took a myofibril, placed it on the glass cover slip, flooded in fluorescent ATP, which is not fluorescent until you illuminate it with light. In this case, it's 561 nanometer light to illuminate the Side 3 ATP. And we, as you can see, illuminate this at an inclined angle. So this reduces the amount of noise, so it improves the signal to noise for our imaging. And what we're interested in are these fluorescent ATP molecules that bind to the myofibril on the surface. And these are the kind of data that we get. So you can see here in purple, on these vertical lines, the alpha-actinin marked Z lines. And so these are marked with an Alexa 488 anti-alpha-actinin antibody. And we can see these little flashes of light. These flashes of light are individual ATP molecules binding to the myosins. And from these data, we can extract both their position and the duration of attachment with very high spatio-temporal resolution. In fact, our spatial precision is 30 nanometers. Spatial precision for our Z line localization is 32 nanometers. So it's fairly high precision. And if we take all of these attachment events and we plot their lifetimes as a cumulative decay plot, we get this relationship, as you can see here. Now, I will point out that this is on a logarithmic scale, and anything plotted on a logarithmic scale versus linear time that is non-linear, i.e. there's a curve to it, tells us there's multiple exponential processes. And so we found the best fit to this to be to 3 exponential processes. And we think that 3 different processes reflect, first of all, rapid interactions of ATP with myosin and or other proteins. And there is evidence for this. Let me just get rid of that. There is evidence for this from the literature. So Mamta from Bernard Brenner's group at the time studied the turnover of individual ATPs and found that there were fast and slow events. And more recently, Alf Manson in 22, Marco Usaj in his group also measured ATP binding to myosins on the surface, and again found that to be a multi-exponential process, with the fast process potentially being the interactions of ATP non-specifically binding to the surface and potentially increasing the on rate of ATP into the active site. The second and third rate constants from this graph here, the second and third rate constants would correspond to the DRX and SRX, and that's because of the rate similarity. But before we can go into rate similarity, we need to talk about how we image, because one of the issues that we have when we're studying any fluorophore is photobleaching. So what we've been doing to eliminate photobleaching is using a stroboscopic illumination approach. And let me briefly explain what that means. So imagine we have a fluorophore undergoing continuous illumination. This fluorophore can only withstand 3 periods of illumination. So each one of these squares corresponds to 200 milliseconds of being illuminated. So if we've got a continuous frame, if we're taking frames continuously, that means after 600 milliseconds, this guy will photobleach. Now, if we take that very same fluorophore and we expose it to 600 milliseconds of light exposure, but this time put a 400 millisecond gap between them, that means that the total amount of time that we've imaged that for is now 1800 milliseconds. And as we increase this time lag, this time interval, in this case 600 milliseconds, we've now extended the lifetime of that fluorophore to 2400 milliseconds. And so this actually turns out to follow a simple linear relationship based on this, this relationship here, which is the fluorescent spot can either disappear through release of the nucleotide or through photobleaching. And so that leads us to this equation that I show you down here. And that is a linear equation when plotted as the observed rate constant multiplied by that time interval on the y-axis, against on the x-axis the time interval again. That tells us the slope of this line is actually the true K-off, and the intercept with the y-axis contains the photobleaching term KBL. So from this we can actually then work out what our expected or what our true off rate is, and from those we can deconvolute these data to tell us that the fast rate constant is 0.36 per second, which matches fairly well with the with Manta and Marco Usage's work, and also the DRX rate constant is 0.04 per second, SRX rate constant is 0.004 per second, which fits in really well with the work from the Cook and Jeeves labs and the work I showed you a little bit earlier. The other thing we need to do to the data is, before we can actually start to get to the nuts and bolts of looking at the amount of SRX and DRX, is actually take into account the fact that fast events are more common. Stands to reason, if something's turning over at 0.36 per second, we're going to see many more of those than we would see as something that's turning over with a rate constant of 0.004 per second. So this is what our population looks like uncorrected. So you have a very large amount of the active population, but if we then correct them according to their rate constants, we actually get the true values of the slow and medium rate constants corresponding to the SRX and DRX populations. So what does that mean? Let's go to look at the sarcomere here. This is a study from 1976 that gives us really nice view of the myosin binding protein C lines in the C zone for rabbit psoas, and that's the protein that we've the muscle type that we've been studying in this study. And so you can see here we have the P zone, we have the C zone, and we have the D zone of the thick filament. And pinching a slide from Paula Bennett's work, you can get to see that this corresponds to all of the repeats of myosin along thick filament. If we then take those data and measure the kinetics for each one of those regions and plot the cumulative residence times for each of those regions, we then get an idea of how the populations of SRX, DRX, and active distribute across those three different zones. Excuse me. So what we can see is that in the P zone, we see a large amount of DRX, whereas the C zone is predominantly SRX, and the D zone seems to be an equal balance of DRX and SRX. And I'll just remind you again, this is in rabbit psoas myofibrils. The active population is sits here, and I'm not going to be talking very much more about the active population because I just want to focus on the SRX and DRX. So in order to be able to study the factors that alter thick filament regulation, we need to look into what those factors could be. Could be, as I mentioned earlier, RLC phosphorylation, myosin binding protein C phosphorylation, force. Now, force is something we can't measure at this point because our myofibrils are laid down on a surface. Calcium would take us out of the relaxed condition because everything we're looking at is relaxed. So those are two components that we're not looking at, and that really leaves us to study phosphorylation. So how does protein kinase A phosphorylation affect distribution in skeletal muscle? Well, let's take a look at what it could do. So, of course, most of what we understand about phosphorylation of myosin binding protein C comes from cardiac, and we're looking at fast skeletal. and we know that fast skeletal has no known phosphorylation sites. However, we do know that myosin-binding protein C in rabbit psoas is about 50% slow muscle myosin, and slow muscle myosin does get phosphorylated. So how does that affect our populations? Well, actually, it affects them quite profoundly. So these are the data that I showed you before. When we add pKa, you can now take a look at these data. And you see here that the SRX has dropped enormously, certainly in the P zone and in the C zone, and it's around about the same in the D zone. Now, just to kind of clarify this, because these are multiple graphs for you to look at all at once, we combine this into an SRX-DRX ratio, and what you can see here is that the biggest statistical difference between the untreated, shown in the solid bars, versus the pKa treated comes about in the C zone. So we see the biggest difference right here. We can do the very same experiment, but with mavacamten this time. And we're working with quite high concentrations of mavacamten because we're working in skeletal muscle. And what we can see here is comparing the untreated, the gain shown in the bars, solid bars, with the mavacamten treated. You see here, the P zone, not much of a difference, but we see an increase in the C zone and an even larger increase in the D zone, so that the C zone and D zone actually end up being approximately the same in the presence of mavacamten. So that's increasing the amount of SRX. So we're getting a really good handle in these experiments of seeing where the SRX and DRX distribute within the thick filament. What's also interesting is we don't see a complete loss of DRX despite using 30 micromolar MAVA. Okay, so those were data that we put together and published at the beginning of this year. And since then, we've moved towards trying to understand beta cardiac myosin working in porcine left ventricular cardiac tissue. And this has been a collaboration with Mike Rainier and Mike Jeeves. And this is the work of Matvey Piligov, who I've spoken about already, and also a master's student, Atika Naim, and a borrowed PhD student from Mike Rainier's lab, Sanette Stetsina. And so working with these guys, they overcame the real problems that we had initially, which were trying to get the sample to a condition whereby we could actually start to image it. And that took us many months of working with the porcine myofibrils. But eventually we got them, and we work. So we, as you can see here, we have a and that just means that we're now right there.
The sound is fading.
Sorry. Cannot hear.
Can you not hear me?
Yeah, I can't hear him anymore.
Okay, just 1 second.
Can you hear me now?
Yeah. Yep.
Okay,
Yep.
Brilliant. I'll go back to where I was. Right, okay. So I should be sharing now. So we're back, I hope. Right, so should I repeat this slide again, or did you get me to the point of 1.7 microns?
Okay, here.
Yeah, 1.7 started 1.75.
Yeah.
Okay, brilliant. Okay, so we worked with sarcomeres of length 1.7 or higher. Okay, these are the data we get. We get really nice myofibrils now. I've switched the color scheme so you can see in cyan the Z lines, and in magenta we can see the individual fluorescent ATPs binding. And to cut long story short, I put all the data together, but I will definitely have to tell you this is very preliminary data at this stage, so it's subject, despite me being recorded, to change. And so what we've seen so far is when we plot the proportion of DRX, we can see that in the untreated, across the different zones, P, C, and D, we can see that we've got approximately the same amount of of DRX, proportion DRX in the across the zones. Now, when we treat these myofibrils with pKa, we actually see a huge increase in the amount of DRX. It's wholly consistent with these with phosphorylation allowing the heads to come off the thick filament. And this would be something that we'd expect to see located on the C zone, which is what we saw for skeletal. But it seems to be across both sides of the thick filament from the C zone, suggesting potentially some kind of cooperativity that emerges from the derepression of the C zone, probably due to pKa phosphorylation of myosin binding protein C. But we've also dephosphorylated using lambda phosphatase, and that's what's shown in these grey bars. So in these grey bars, you can see that there's actually a reduction relative to untreated, probably less so in the P zone, but there's a reduction relative to untreated in the amount of DRX. So we're starting to see more SRX when we dephosphorylate, and the dephosphorylation will include dephosphorylation of RLC, myosin binding protein C, TNI, and titin. And so that complete loss of phosphorylation really kind of seems to shut down the thick filament. And interestingly, if we then take a look at mavacamten treatment of the original untreated stuff, so we haven't added any other phosphorylation or dephosphorylation to this. If we just look at mavacamten effects, we can see mavacamten here actually also reduces the amount of DRX, so increases SRX in that case, but to a very similar level of the dephosphorylation. It doesn't go all the way down to zero, and we're using a lot of mavacamten here. It doesn't go all the way down to zero. In fact, hovers around about the same value as the dephosphorylation, suggesting that maybe dephosphorylation wouldn't take us any further, but we have yet to do those experiments. So the other nice thing that we can do actually is we can increase our resolution. So far I've only been talking to you about studying things in the P, C, and D zones. But what we can actually do is decrease the size of our bin. So we dropped our bin sizes down to 50 nanometers and looked at the, in this case, frequency of events. And so what we've actually done is we've colored in the DRX and the SRX in blue and black. But I'm not going to ask you to pay too much attention to this. Again, this is preliminary data, and we haven't fully assigned that, but our initial assignments show the distribution here. But the important thing to see here is that in the C zone, you can clearly see there is a reduction in the frequency of events, telling us the activity of the C zone in these relaxed cardiac myofibrils is much lower. And in fact, you can see here that there's potentially even a ramping up this way, probably less so that you can see here, but certainly a ramping up this way that suggests there may even be some cooperativity emanating from the C zone, which kind of fits a little bit with what we saw with the pKa data. So it clearly shows that we have a reduction in ATP in the C zone, and we're hoping to go with get even higher resolution soon. One last thing I'd like to talk about, which is where we're moving forward to. Right at the moment, we want to go beyond relaxed myofibrils, of course, and this it kind of emerged from a really great trip we had to Florence, to Italy, to meet Corrado and Chiara, and they essentially taught us how to build one of these systems. Let's play with their system and have a look. And so here is an inverted microscope with our microneedles that enable us to capture a single myofibril, and we've now equipped this with fluorescence imaging. And you can see in this lower image here that we're now able to actually see individual Z lines fluorescently tagged using that same alpha-actin antibody suspended between these two microneedles. And so you can see here along that line that I've now just shaded in unhelpfully, you can see the Z lines. And this is the first step. This is a low resolution image. We're moving this up to high resolution soon. And hopefully what we'll be able to do is actually work in active conditions where we've fixed the ends and are looking at the activity of the myosins. And this would really complement some of the work that we heard from Elisabetta's group earlier. And I think this would be a really exciting next step for what we're looking at. So just to conclude, for skeletal thick filaments, we've shown that PKA activates heads from the SRX, but it does it largely from the C zone. And that indicates that myosin binding protein C in skeletal is phosphorylation sensitive, kind of like cardiac myosin. Mavacamten depresses activity throughout the thick filament, and we see a little bit more in a D zone because there's probably more it can repress, plus we don't see it going to zero. So it doesn't completely repress activity in the thick filament. For cardiac, we see that phosphorylation seems to stimulate across all zones. Dephosphorylation drops activity to the level that we see when we treat with mavacamten. And we're treating with about 30 micromolar mavacamten for cardiac, which is a huge amount. And again, we don't see full loss of activity. And high resolution imaging suggests that we might be able to get down towards the crown level soon. And so that just leaves me to thank everybody who's been involved in this work. So Johnny Walklate, Matt Pilagov, and I'll thank these people as I won't accept Johnny Walklate, of course, but Johnny's been involved in looking at the kinetics and underpinning some of the imaging that we've been doing. Lawrence Helling, a master student involved in this, Atika Name, who as data went into that high resolution imaging, along with Sonet's data, Sonet Stetsina from Mike Renier's lab, and also a collaboration with Mark Jeeves. And I'd like to thank the School of Biosciences for funding this, as well as NIH, Royal Society, and Horizon 2020. And I thank you for listening. I'm happy to take questions.
Very, very nice talk. Thank you, Neil. Very exciting result. Yeah, if you have questions, you can unmute yourself. There's one in the chat if you want to take it.
Okay, I'll take a look at the chat.
Yeah, I can actually ask in real time.
Yeah, please do.
Yeah, great results. Thank you. It was not clear, were these myofibrils from the main set experiments for generating, or, I mean, I know
so all of these were in relaxed conditions.
Only relaxed condition.
Only relaxed, yeah, absolutely. And so that's where we're hoping to move towards. We'd really like to be able to, let me just get out of this. I think I discard that. We'd really like to move towards, sorry about this. We'd really like to move towards actually studying these things directly. So with force, having microneedles on the ends of these myofibrils.
Right.
And that was that last image I showed where you could see the Z lines. We're moving forward to try and get single molecule resolution there, but it is a lot more tricky because now we're working in conditions above the surface where also got a lot of noise.
So you plan pure isometric condition, right? So it will be not bending.
Yeah, yeah.
So how are we going to measure force in this case? It's kind of like,
well, so we're just going to do fixed end measurements straight off the bat. And we can measure force because we've got the microneedle system, so we can look at the bending of the needles in the same way that the Corrado's group's been doing for a number of years. So we have the capacity to do all of that, and also measure the sarcomere lengths in parallel because we'd be picking those out of the Z lines.
Thank you.
I would suggest you're going to have a limited amount of time when you activate the myofibril because it's not that solid of a prep. But I think you can probably get relatively long recordings at, partial calciums. So yeah.
I think that's good advice. I think you're absolutely right, Mike. We talked about this before. I think working at submaximal calcium will give us a longer lifetime for the myofibril. Yeah,
and you've also got to find your focal sweet spot after you activate and gets into steady state. So I would just suggest that it's going to be a Herculean effort, and if anybody can get it done, it'll be Neil.
Too kind.
I have a question.
Hi.
There's still time. Yep. Hi. Nice talk. I was actually just reading the one of your papers like 2 days ago by chance.
All right,
Really
you're the
cool
one.
stuff. Gets me very excited. Yes. So okay. You have, it looks like you have sarcomere length information along with this other details about P zone, D zone, C zone. Have you tried to do some sort of sarcomere length relationship to see if there's some changes, or is the variability too much to?
I think we're limited by a number of factors there. So the issue is that for each sarcomere, we're only collecting a couple of events. And so if there's sarcomere to sarcomere variability, which there will be along the myofibril, if we're trying to assign any kind of sarcomere length dependence to individual events within a sarcomere, we just don't have enough data. So over a 30-minute recording, we'll get around about 200 ATPs. And so we have to record for hours and hours and hours. So what we do is we refresh myofibrils regularly to kind of keep them as fresh as possible. We then take all of those data, put them together, and we have to aggregate those together. The one thing that will address exactly what you're suggesting is what I've just answered in the previous question, which is get those things onto a myofibril rig and then stretch them out. And then we can do a sarcomere length dependence that way.
Nice. Thank you.
Thank you.